Good afternoon. My name is Bill Detmer, CEO of Unbound Medicine. Thank you for joining us today as we get the latest on a topic that has dominated our world for the better part of a year, COVID-19. We're very lucky to have today with us our friend, Dr. Paul Auwaerter, who has been at the center of this work for the last year. He's professor of medicine at Johns Hopkins University School of Medicine, serves as the clinical director for the Division of Infectious Diseases and Center for Environmental Infectious Diseases, and has served as past president of the Infectious Disease Society of America, IDSA, the largest organization serving infectious disease professionals worldwide. So we're delighted to have Paul, a friend of Unbound Medicine, working together on the Johns Hopkins Antibiotic Guide and other Hopkins guides for over 10 years. So today's presentation will include a question and answer format, some of which Paul and I have discussed and arranged in advance, and others were submitted by you. And so your questions will be posed and answered by Paul as we move through this presentation. So thanks again for attending. So Paul, let's just start out with an update on transmission. What have we learned over the last few months?
Yeah, Bill, thanks very much for the introduction, and delighted to be here today. And what's different from the past is, I think, as we're moving ahead, we've already seen progress, and the hopes are, despite numbers worsening, I really hope in 2021 that every month gets better. But I think there's been general consensus, Bill, that one of the ways this virus is so successful is that people who are unaware they're ill transmit the virus. And really, I think the best estimates I've seen, including from Tony Fauci and the NIH, are that approximately 40% of spread is accomplished by people that are unaware. And this is because we've clearly learned that people are highly infectious 1 to 2 days, even before they have symptoms, for the people that do develop symptoms, and then remain so for perhaps 5+ additional days. So you have 1 week where people are highly infectious. And although super spreader events and other things have really gotten a lot of attention, most illness is actually acquired at home where people are living. Now, compared to early on, when in the spring there was much chaos and uncertainty about this novel coronavirus, mortality rates have diminished significantly. I mean, they're still much higher than seasonal influenza. But for example, in this paper from New York City academic hospitals, over 25% total mortality rate in March and April, fell to 7.6%. And I'll tell you, we had similar numbers at Johns Hopkins in April and May, but really in September and October, we're now between 5 and 7% overall. So the big improvements. Now, why is that? I think we're not sure. There are certainly trends where people are now younger. Initially we saw many people in nursing facilities and so on, but there are other factors, Bill, here too.
Great. So in terms of therapeutics, obviously this has been an amazing year of science and the application of that to clinical medicine, learning about this novel coronavirus and then applying principles, learning clinical trials out to the periphery. What have we learned? We'll talk a little bit later about specific therapeutic modalities, but what have you learned about the general approach and how has that influenced mortality?
Yeah, so I don't think we really know, Bill, all the details. So we already talked a little bit about the shift to younger patients, but I'll throw out some issues there. Now, first, compared to the spring, more people are wearing masks, social distancing, and there is a sense that if people are exposed to a lower viral load, then it takes more time to make a lot of virus, and that, of course, gives the immune system more time to react. So that's one theory. The other is that we're now dealing with patients that are not waiting until they're short of breath for presentation. Certainly, we know much more about ventilator management and even proning, which is very helpful for ventilating patients without the need to go on ventilators. So I think there's a lot of early ventilator management that's now not nearly as common. And we'll talk about treatments in a moment, but dexamethasone is the only one at the moment that clearly has a mortality benefit that everyone agrees upon. But Bill, mortality is not the only issue. Obviously, with the strain on hospital systems, trying to prevent patients from getting or requiring the hospital, as well as trying to get them well and discharged from the hospital, are now critically important.
So as we think about therapeutics, there are obviously many different approaches to mitigating the illness once it's acquired, and at different locations, at home, pre-hospital, early hospital, and obviously when people get severe COVID in places like the ICU. Can you talk a little bit about what we've learned about the general approach to that trajectory of a typical patient?
Yeah, so Bill, just briefly, certainly what we know about COVID-19 for people that are ill, there is an early viral phase, and the people that get more ill look like they have a hyperinflammatory response. And then perhaps the most dire who become ill develop a lung injury syndrome or multi-organ system failure, where people can be in the hospital for weeks and weeks. Now, just in brief, certainly the NIH, and there are other groups such as the Infectious Disease Society of America that have developed treatment guidelines. And the most recent update from early December, I think, sort of highlights some changes that if you've been caring for patients with COVID-19, I can just try to synthesize a bit for you. So, for example, if people are hospitalized but not requiring oxygen, at the moment there's no clear evidence, although Remdesivir is FDA approved for this group. That's the blue bar. I would say most patients fall in the gold bar where you need oxygen. And interestingly, with the release of the final Act 1 trial, which compared Remdesivir to placebo, they initially were graded as an A1 recommendation. That's been downgraded to B2A, so a bit less, and so only moderate levels of evidence to suggest that the antiviral Remdesivir is helpful, along with anti-inflammatories, which are more important, especially in the gray bar, in people that are critically ill. So for the moment, if you look at this, it's really Remdesivir and dexamethasone that depends. With the best grade of evidence is the recovery trial, where the more critically ill patients seem to definitely benefit with less mortality due to dexamethasone. So Remdesivir, dexamethasone, at least for our hospitalized patients, seems to be the standard of care. Now, I'll mention, of course, in Europe and people that are following the WHO Solidarity trial, Remdesivir there did not have any impact. But Bill, I'll be honest, I thought the Solidarity trial is not the way to evaluate drugs. There was no placebo arm. And, I think there's legitimate critiques about how solid that data might be in terms of not having an impact in late presentations, and certain countries perhaps being overrepresented for some treatment arms.
Excellent. Well, now maybe we could dive into some of the therapeutic classes. We've obviously, you've talked about Dexamethasone and Remdesivir. Other classes, for instance, antibody-based therapies have gotten more attention recently. With when our president got ill, he was a recipient of one of the cocktails. Could you give us an update on how beneficial it is and at what point in the illness it might be most beneficial?
Yeah, so there's certainly a lot of biologic plausibility for antibodies to help in an acute viral illness. By binding a virus with a neutralizing antibody, the virus can no longer productively infect cells and therefore really mitigating illness. Now, of course, you'd love to have evidence that that's truly so for SARS-CoV-2. And so there are two products that have now received emergency use authorization, which of course means there's still investigational investigational products. But the first was Bamlanivimab, which is a single monoclonal antibody directed against the spike protein, the protein that docks to the ACE2 receptor. And this BLAZE-1 trial only looked at outpatients who had mild to moderate disease. And it was a dose-ranging study that was thought to look at viral endpoints, but that wasn't met. But the FDA thought that based on a secondary endpoint of avoiding an emergency room visit or hospitalization within a month of administration was important. As you can see, it did meet that role, but they decided to limit it only for patients at high risk for severe complications. So people with comorbidities, advanced age, over 65, for example. And so the EUA has that embedded in it. But the information is rather slight. And in fact, in this New England Journal paper, we don't really know of the 3% or 10% how many were emergency room versus a hospitalization. And if you look at the group here that was studied, you can look at the demographics, but about 70%, 60 to 70% did have risk factors for severe COVID-19. And about a third had considerable obesity, for example. And in terms of clinical benefit, did patients feel better? You know, there was perhaps at the medium dose, which was not the FDA approved dose, some clinical improvement around day 3 to 4, where patients felt a bit better with no overlap in the confidence intervals. But when you actually pool all the 3 different doses, that still held up in that modest range. So not something that you'd really feel is worth a 1-hour infusion and 1-hour follow-up and so on and so forth. But the FDA elected to choose the 700 or the low milligram dose, which was generally well tolerated. And this would be true, I think, for most biologic agents. There are many monoclonal antibody agents out there, and they're generally very well tolerated in terms of any infusion-related reactions. So this is not a surprise, but this was a key outcome here, again, where it was 6.3% of people hospitalized versus only 1.6% in this particular group here. So the benefits seem to be most, or at least the patients were older, who were also had a higher BMI. And it's likely that this drug is best if given early. Now, when the drug was given later in hospitalized patients, there was worse outcomes of concern. So the trial called ACTIV-3, which was in again, a federal study, was halted in this regard. So this was the first one released, and at least at Hopkins, we didn't have enough information to make a formal recommendation, and we considered that there is still an outpatient trial where rigorous data can be gathered. So that was our recommendation. The NIH and IDSA both thought there was either insufficient data or there wasn't enough to really make a recommendation for routine use. Now, I'd have to say if I had a high-risk patient, someone who's quite elderly, someone that has multiple comorbidities, I'd give some thought to using it, but there's not a whole lot of evidence, Bill, to back that up. The product that the president got in early October is Casirivimab plus Imdevimab, two anti-spike monoclonal antibodies made by a different company that had a somewhat larger trial with a set dose, looked at a number of factors, but it too appeared to have about a 60% reduction in medical visits, and this was maybe a little higher in people with comorbidities. And most of the benefit, which is not surprising, were in people who presented early enough, they didn't have measurable antibodies in the blood, and that's usually before day 6 or 7. And there were few adverse events. So this also received an EUA identically for outpatients only with high risk for complications. Less than 10 days, but to be honest, I wouldn't even think about it in anyone that has 7 days or more. So it would have to be less than 7 days. It too, for hospitalized patients, had its trial halted for the time being, although it may resume under different auspices. So these are functionally, Bill, the only two outpatient products at the moment that have therapeutics for COVID-19.
Thanks for that. Thanks for that update. That's very good material. Let's turn now to maybe an old friend being used in a new disease, convalescent plasma. This has been not really risen to that much awareness, but of course is an old approach to therapeutics. Could you talk about its role, if any, in the current situation?
Yeah, so Bill, I think convalescent plasma for patients that recovered was really embraced early on as much like we thought monoclonals might, but with much greater availability for treatment of early infection. Now, this obviously is not a single antibody, it's a complex product. And a lot of this was based on a small experience with the original SARS, where it did look like people were able to have better outcomes if they had gotten earlier use of the plasma there, and whether they were PCR positive but seronegative, again, suggesting earlier treatment seemed to benefit when they used convalescent plasma in the original SARS. So it may not just be neutralization of virus. For example, there could be other impacts by controlling hyperinflammation or even some impact on the hypercoagulable state. But there's certainly data early on from New York City, which was a well-done study with matched observational controls that suggested that if patients had gotten plasma before they required higher levels of oxygen, that they had improved outcome, so that they needed less oxygen and even had a improved survival. And that's only for patients that were not yet in the intensive care unit, again, suggesting earlier treatment. What happened, and this is a bit of a complex story, the FDA approved what's called an expanded access program. So even without data, the thought that this therapy might work meant that blood banks across the country found patients who donated plasma. The Mayo Clinic helped coordinate this. A very large study, a subset of a little over 2,000 patients, though, have been studied. This is still in preprint, has not yet been through complete peer review, but this is how what's now an EUA for convalescent plasma was thought to be derived, in that if patients got it within 3 days of their diagnosis, there was a trend towards lower mortality rate that held up even at 30 days. And then in terms of biologic plausibility, the mortality rates were lower in people that got high titered plasma versus low titered plasma, Bill. So now, as of early December, any plasma product has to be labeled high or low titer by the blood bank, so clinicians and patients both know what kind of product they're getting. You can still give a low titer plasma, but you have to make sure you inform the patient if there isn't any high titer plasma available. Now, there have been a number of trials that have not really shown a lot. The one that got a lot of press was from the early experience in Wuhan, China, but the epidemic there really stopped, so they didn't fully enroll. So this was a negative trial, but there was a sense that patients did a little better if they had severe disease, but not critical disease, so early on. Most recently, I'll point out to two trials from Argentina that really show the concept that if you're a clinician and want to use convalescent plasma, give it early. This well done randomized controlled trial in the New England Journal looked at patients with severe COVID-19 who on average got it at day 8 of their illness, and it did not seem to be any substantial improvement in terms of outcomes or mortality at 30 days, so a negative study. By contrast, and I didn't include this slide, there is a preprint of another Argentinian study from that gave plasma to elderly patients, Bill, but they had to have less than 3 days of symptoms, and that did have an effect on outcomes and mortality. So I think as this remains an investigational agent, there are a number of large studies in progress regarding convalescent plasma. The recovery trial has a component of this. There's a New York City trial, as well as one that's based in Nashville. So I think in the next few weeks to even months, we'll have information for hospitalized patients that's more robust. Also, it's being explored for both prevention and treatment of outpatients as well, much like the monoclonal antibodies.
Excellent. Well, before we move on to vaccines in a moment, let's cover one more area of therapeutics, and that would be the immunomodulators. There's been interesting work there. Can you fill us in with the latest?
Yeah, so early on, I think there was a lot of hope that targeted therapy against cytokines such as interleukin-6 might be helpful in these patients. And the model was that it might be like a CAR T cell cytokine storm that you see in that particular oncologic treatment. But although the Chinese guidelines had Tocilizumab based on that premise as part of their guidelines, there really has not yet been convincing benefit. A number of RCTs have been negative. One that's, at least by press release, said to be positive. The IMPACTA trial was done in different countries. We're still waiting to see that data in peer-reviewed form. And there's another one that's said to be positive, but Bill, the data is quite mixed on Tocilizumab. So compared to earlier in the epidemic, I would say people are not really using this to any degree. And Dexamethasone remains the sort of immunomodulator, as it were, of choice. Now, the second trial from the ACTT, the NIAID trial, looked at Remdesivir versus Remdesivir plus a JAK1, JAK2 inhibitor, Baricitinib, which is FDA approved for rheumatoid arthritis. And so this is, again, a very well done trial, just recently released last week, of over 1,000 patients. And a couple points of interest to me, at least, was there was only a 1-day difference when you added the Baricitinib compared to placebo plus Remdesivir. But remember Act 1, where the Remdesivir treated people got out at 10 days in terms of time to recovery versus 15. So it just shows you how care has improved overall. But the 1-day difference, I think for most people, you're wondering, would it make a difference? It's a relatively expensive drug. However, there was one group, if you dive into this paper, the so-called ordinal 6 group, who needed high flow oxygen, not low flow, that got out of the hospital, I'm sorry, it's the wrong term, recovered 8 days faster than those that did not get the drug and only Remdesivir alone, which I think was incredibly impressive, although was a subgroup analysis rather than a primary endpoint. So at the moment, it's unclear how this might be used. Personally, I might use it in someone that can't take steroids, perhaps someone that's in DKA or something like this, along with COVID-19. I think this will be answered by a trial that's just launching called ACT4, which is Remdesivir and Baricitinib versus the standard of care right now, Remdesivir plus Dexamethasone. And it's only going to be looking at patients earlier in illness, so non-ICU, I believe, ordinal 6 and before. So, but we're not going to have this information for several months.
Mm-hmm. Fascinating, fascinating. Well, let's go on to vaccines. This has been an amazing story within this larger story. We're here in December, about a year since the virus was first identified, and just in the last couple of weeks, we've had the Pfizer BioNTech vaccine approved in the UK, Canada, and the US under emergency use. Could you give us an update of where we are and where we're likely to go in the next few months?
Well, I think you'll be reading about it in the papers quite a bit in terms of distribution plans. I think everyone knows that most states are following the CDC, ACIP, and National Academy of Medicine recommendations for getting healthcare workers and frontline workers immunized, and then there'll be high-risk groups, people living in congregate living, and so on. You know, the mRNA vaccines, I have to say, I do not have qualms about using, despite the fact that these have never been used previously as a vaccine approach. The way how I like to describe it to patients are that this vaccine and also the Moderna vaccine, which is in front of the FDA now, are very similar, where unlike a virus where you'll have 8 genes or more of RNA, here you just have one. And instead of a viral envelope, you have a lipid nanoparticle. So you really have a mini virus, as it were, and a virus that's incapable of self-replicating. In fact, the RNA degrades rather rapidly, so and is not something that integrates into your genomic material. So I think so far the early data suggests that these approaches are very safe. But there's a little schema here, Bill, that shows that for an RNA vaccine on the right-hand pink part of the slide, you can see once the liposome merges with the cytoplasm, the RNA is processed through the endosome. It's then, of course, transcribed and translated, as it were, to produce the spike protein, which is then presented with a MHC molecule, and you'll get your immune responses. Predominantly, we think more humoral related with the idea of making neutralizing antibodies, but T-cell responses are also being studied as well. So that's the concept, and at least the Pfizer vaccine from the FDA brochure that was submitted last week there looked highly effective, as you can see here, 95% overall, slightly less in people that are over 55, slightly higher in people that are younger, but really overall fantastic. And if you look at the graph about cumulative incidence, what's also remarkable is that the first dose alone seems to stimulate this protection. So the second dose, I think, is really to help stimulate longer lived immunity, but people appear to develop some protection within 7 to 10 days of that first jab. So this is just a remarkable achievement. I think many of us were surprised at how effective this approach is. The Moderna vaccine looks very similar, and that's in front of the FDA. Many people are worried about side effects. I think the upshot, I say, is that if you look at this side by side, actually the new shingles vaccine that many of us have gotten actually is about the same to a bit worse. There's a lot of local reactions, and you might be sort of ill for a day or two as a consequence. But importantly, in terms of serious side effects, there are really none that seem to be concerning. And so, Bill, overall, I know there were the two people in the UK with a history of severe allergies that had anaphylactic type reactions. So I think this is not uncommon with any new product. Some people are going to react, especially when we're giving it on a mass scale. It's interesting that in these nearly 40,000 patients, that wasn't witnessed in the clinical trial, but of course, patients with a severe allergy history were excluded. So that's perhaps the only cautionary note that I know of, and that information was incorporated into the EUA document and the requests of the ACIP. So that's where we are. And of course, we try to keep our SARS-CoV-2 module updated every week or two, if possible, as new information comes to light. And I think we're going to get to some questions.
Excellent, excellent summary, Paul. Thank you very much for that. We've gotten a lot of questions from our friends and associates and colleagues around the country, and let's dive into some of those. A lot of them have to do with the vaccine, as you might expect, since that is top of mind. The first one is there any significant concern that any of the current vaccines could actually induce some sort of immune response that could be deleterious, either short or long term?
Yeah, so far, at least for the spike protein-based mRNA vaccines, it does not appear to be the case, but the full safety signal is going to take until late spring or summer to see at the end of 6 months. So, early on, though, I'd have to say, usually you see most vaccine adverse reactions within 4 to 8 weeks after starting a trial. And those have not been seen, Bill, with this. So I feel fairly confident if I'm able to get the vaccine, since I'll be on a COVID ward working in January, I will take this, either the Pfizer or Moderna vaccines. Now, the ones that are coming up, I think, are a little different. For example, the Oxford AstraZeneca vaccine is one that's probably furthest along, although probably won't be in front of the FDA because they are behind in their US trial. But, there have been some cases that I think were MS, and there were concerns about this. And with any adenovirus, a concern that even though this isn't replicative, that there might be more cross-reactivity, induction of autoimmunity. So I think we'll wait for the full data set. I think the information that was presented so far on this chimpanzee adenovirus vector was very confusing to read, and I don't think inspired a lot of confidence.
Mm-hmm. Yeah. Related to that, people are interested in what you should do if you've been exposed, you think that you've had COVID, or even if you've gone on to be shown that you've were tested with the antigen test or later with the antibody test positive, should you get the vaccine or not?
Yeah, so I have a couple of thoughts on this. So I think if you have a confirmed case of COVID-19, so a molecular test or an antigen test, I think I would divide it into two sets of people. If you had a severe illness, we know that tends to engender strong antibody responses. And so you're probably pretty well protected, we think, for 3 to 4 months, at least, if not longer. So I'd rather have other people get that vaccine, but I don't think there'd be a danger if you elected to go ahead and get the vaccine. And at some point it would be useful because you could then have a better knowledge of vaccine impact and durability and protection for yourself.
Mm-hmm.
For people that are asymptomatic, but got COVID-19, we know those people tend not to develop as strong antibody responses. So I think those people I would certainly recommend immunization. Now, for people that are only antibody positive, they got a test. The question is that really because they were exposed to the novel coronavirus, or is this because they had other coronaviruses earlier? In that group, many were in the Pfizer and Moderna trials, for example. So we knew that there were, I think, 600 or 700 in each arm in one of the trials that had pre-existing antibody levels. So there, I would recommend going ahead and getting the vaccine. I don't think it caused any harm. And I think it's hard to know whether you know, especially if you didn't have symptoms you thought were consistent with a coronavirus infection, what those antibodies mean at this time.
Mm-hmm. Excellent. Let's talk a little bit about the demographics of the participants in these trials and the representativeness of our society. That's been a point of contention for years about clinical trials, whether they represent the groups for which the agent ultimately would serve. And more to the point, are those groups well represented in these trials? And is there any indication that the vaccine is more or less effective in various demographic groups as well as age groups? And how would that affect the rollout of the vaccine nationally?
Yeah, so I think efforts were certainly made to recruit, certainly in the United States. So people of color, for example, elderly, who are normally excluded, right, because they don't react as well, but those are the people at greatest risk. So I don't think it's quite as reflective of what we think American society represents. But I think this is a very sensitive issue, and I know from a vaccine distribution standpoint, there are many discussions about trying to make sure people that are most at risk are recipients of this vaccine. So there'll be a lot of education. Do people that are understudied have different immune reactivities or side effects? I think that hasn't come up. I'm not sure we know enough about that yet, Bill. I think we can always try to do better, but it's been difficult to have as many people in the Latinx community, Blacks, for example. We love to have more trial participation. I think there are certainly historical reasons why that's not as strong as we'd like or as easy as we'd like there. So all, I think, highly important scientific and also societal questions.
Mm-hmm. How about kids? So notice that the trials have not included children under the age of 18 or 16, depending on which trial we're talking about. And then the EUAs have been only for adults, essentially. What do you think about children and vaccination and the likely steps that we need to take to know what to do and when to do it?
Yeah, so you know, those are interesting, and I know the Moderna vaccine has already started its pediatric trials. You know, the vaccines at the moment are still investigational, so they really can't be given to children. But we certainly have, I think, we're beginning to understand correlates of protection better and better. And it may be that instead of having placebo-controlled trials or so on in children, we'll be able to just move to surrogates. This is certainly true for other vaccines that have become established. So children, I think, are going to be important and necessary, mainly because I'm sure they do transmit infection, especially adolescents, perhaps more than elementary school children. Some children, of course, do get severely ill. It's much less common, so it's not quite as much a priority. It's also clear that unlike influenza, where school children are a prime group that spreads influenza infection. It's probably less so for children, so the urgency isn't quite as great, but it's very important. I think we understand this group as well because we're not going to achieve 60 to 70% immunization in our country if we don't include children.
Mm-hmm. Most of the vaccines under development have a 2-dose regimen, typically 28 days apart. Several that have not yet reached, have not reported out their phase 3 trials are 1 dose. And you gave pretty interesting results there showing the immunogenicity, how powerful it is even in the first dose within the first 7 to 10 days. What are your thoughts about the kind of interesting intersection of policy of having multiple doses? Well, let's talk about the fact that there's limited number of doses available in the beginning, and trying to get the maximum bang for the buck, if you will. What are your thoughts about that?
So I think again, people were surprised that a single dose yielded protection. So there could be that argument, but as an investigational agent, I think regulatory agencies are appropriately hesitant to mix and match or alter what they're trying to follow at this stage. So I don't think we'll see that expanded in the sense quickly that we'll be giving people 1 injection and then just wait months later to get a second, because we just won't have any data to really assure people that that's effective. The 1 jab vaccines, Johnson & Johnson has one, it's an adenovirus, and those are generally done mainly because of the concerns that when you use certain vectors, pre-existing immunity may limit responses of a booster. So almost all the vaccines being studied are to an initial shot and then your booster coming, as you said, about a month later. So I doubt that will change at this stage. I think there's also a trust and assurance issue here as well. So it's quite a legitimate question just for trying to achieve maximal good versus safety and durability at this stage.
Mm-hmm. So could you just review maybe one of our last questions, sort of the waterfall for how the vaccine might be distributed based on kind of risk and societal needs?
Mm-hmm. Yeah, so certainly it looks like healthcare workers and those who are in those supportive roles are getting the first doses. And then, people at high risk for disease, especially living in congregate facilities or institutions, nursing homes, and so on, then we'll sort of scale down to include people that might be frontline and essential workers, and certainly people that don't have risk factors, otherwise healthy, younger in age, are going to be last in line. The time frame for all this, I think, had been optimistic to try to achieve this, but even if vaccine production was sufficient enough that we had all the doses available, Bill, the logistics of administering 250 million doses times 2 for the most part is really daunting, and I think still needs much to be worked out. So people ask me a frequent question, so when will this all be over? And I said, well, there's some great successes here, but I think you're still looking towards the latter half of 2021 so you get enough people immunized, and then it also depends a lot on vaccine acceptance and by how many.
Mm-hmm. Yeah, and that's a frequent question people ask, which probably won't be known until we have more results from these trials, the long-term outcomes, but how long protection might last from the vaccine. From other vaccines, have we learned, are there lessons that we've learned that could help guide what's possible there? Or anything about the both humoral or cellular immunity of this particular virus point to how effective it will be and for how long?
Yeah. Well, here's a couple of things that I'll mention. So regular coronaviruses, the four that cause, usually the common cold and so on, it appears that immunity lasts probably no longer than a year. With the original SARS infection with the first coronavirus, you could see probably protective levels of neutralizing antibodies for 3 to 4 years. But of course, that infection went away. We're still under a year since the first people were infected for the most part. So we really don't know those answers. And Bill, we know some people have been reinfected. It seems to be rare, but well documented. But this isn't surprising given the, hundreds of millions of people that have been infected. So we don't know for sure, but my sense is from a vaccine standpoint, this is an unanswered question and will only be followed with time. From a natural infection standpoint, Florian Kramer has some interesting studies following a group of people in New York City where it does look like neutralizing antibody levels remain robustly elevated for most people who have been sick, even with mild illness, that seem to be lasting, after some initial decline quite a while. So my bet is that if you had been sick with the coronavirus, you probably have 3 to 4 years of protective infection, at least protection against infection. But of course, we'll be learning more over the next few years about many of these important questions.
Mm-hmm. Well, Paul, we'll conclude with that, and want to thank you so much, not just for today and this awesome information that you provided us, but the leadership of you and your departments, the Department of Infectious Diseases and the larger group at Hopkins, what you all have done for the world really in the last year, and I'm sure will continue to help lead us in the coming year. We're obviously delighted, Unbound Medicine, to be in partnership with you in transmitting some of this information with the Hopkins guides, and particularly the antibiotic guide, where you have been faithfully updating the COVID-19 and the Remdesivir and the hydroxychloroquine and topics, as well as some of your colleagues actually doing wonderful topics on some of the mental health issues with COVID, et cetera. So we're just very thankful for the dedication that you and the organization have shown to all of us citizens and friends like Unbound Medicine. So thank you for that. And if you have any questions about any of this material or about the Hopkins antibiotic guide, please reach out to us at Unbound Medicine at the email webinars@unboundmedicine.com. Thanks again, Paul, and to all of you, have a wonderful day.
Thank you, Bill.